You know, it's genuinely surprising. We often talk about cancer cells just craving sugar, right?
Yeah, like they're just glucose monsters.
Exactly, gobbling it up for that fast growth. But it seems there's another major fuel in the mix.
That's right. And understanding it might really shift how we think about tackling the disease.
So today, we're diving into some, well, pretty groundbreaking new research. It shines a light on a really key metabolic process in cancer.
hmm And this isn't just, theory. We're talking about findings from an actual new paper.
Yeah, and we're using Professor Thomas Seyfried's insights from a recent presentation he gave on this very study as our guide.
He's been a major voice in this area for a while.
Definitely. So for you listening, this is really about getting a clearer view of cancer's basic mechanics, maybe cutting through some of the noise out there.
Right, getting a fresh look at how these cells actually thrive.
So our mission basically is to unpack this new research. What does it really tell us about how cancer cells get their energy, especially regarding one specific amino acid?
That seems to be playing this unexpectedly huge role. Yeah,
Okay, so let's get into it. The researchers started with a pretty straightforward experiment, didn't they?
they did. They took glioblastoma cells. That's a really tough brain cancer, both mouse and human
They
cells.
just starved them.
Essentially, yes. Put them in a basic saline solution. No nutrients, nothing.
Okay, just to see what happens.
Exactly, to see how long they could survive. And right away, something interesting popped up.
Oh, yeah. What was that?
Well, the mouse cells, which generally have a faster metabolism anyway,
Yeah.
they only lasted about 24 hours.
Go.
But the human glioblastoma cells, they held on much longer, like 48, even 72 hours.
Wow. So even baseline survival is quite different.
It really highlights these inherent metabolic differences,
Yeah.
even between cancer cells from different species.
Right. So they've got this baseline, no food survival time. What's next?
Well, the logical next step was to start adding potential fuels back, but selectively, one at a time.
Okay, like different amino acids?
Exactly. They added individual amino acids, sometimes with glucose and sometimes without, just to see what, if anything, could keep those cancer cells going.
And did anything work?
Oh, yeah. And here's where it gets really interesting. One amino acid just blew the others out of the water.
Which one?
Glutamine. When they added glutamine, the cancer cells just took off. A huge boost in growth compared to all the other amino acids they tested.
So glutamine is like super fuel for them.
It really seems to be uniquely powerful for these cells.
What about glutamate? I know that's related.
Good point.
Ah,
Glutamate is the first thing our body makes from glutamine. And they did see a little bump in growth with glutamate.
but not like glutamine itself.
No, not even close.
Yeah.
The glutamine effect was way more dramatic.
Okay. And what happened when they put glucose and glutamine together?
Well, that's the kicker. They seem to work together synergistically.
Meaning like 1 + 1 = 3.
Kind of, yeah. It's like glucose provides some things, glutamine provides others, and together they give the cancer cells everything they need for that really rapid, uncontrolled growth.
So it's this combination that's really driving things.
It looks that way.
Hmm.
This dual fuel situation seems particularly potent.
Okay, so this raises the big question then. How are the cells using this glutamine?
Exactly. That was the core of the study. Are they using it through respiration? The normal way cells use oxygen in the mitochondria to make lots of energy.
Oh,
Or are they fermenting it, using a different pathway that doesn't necessarily need oxygen?
right, like how they ferment glucose into lactate.
Precisely. So is glutamine also being fermented? That was the crucial question.
How did they test that?
Okay, so first experiment to tease this apart.
Okay.
They took away the glucose entirely,
Okay.
but they made sure there was plenty of oxygen available. And they fed the cells only glutamine.
And what happened?
The cells survived. They could tick along using just glutamine and oxygen.
But that doesn't tell you how they were using it, right? Could still be respiration.
Exactly. It didn't distinguish between respiration or fermentation of the glutamine, just showed they could live on it with oxygen present.
So they needed to force the issue, take away the oxygen too?
That was the critical next step, the pivotal experiment, really.
Okay, tell us about that one.
They created hypoxia, basically, an oxygen-free environment.
Okay.
And again, no glucose, only glutamine.
So no oxygen, no glucose.
Yeah.
Just glutamine. What would you expect?
Well, conventionally, you'd think energy production would plummet, right?
Hmm.
No oxygen for respiration, no glucose for fermentation,
Uh-huh.
but they still detected ATP being made,
Ah,
the cell's energy currency.
seriously? Without oxygen?
Yeah, not as much as with oxygen, definitely reduced levels, but it was still being produced.
Whoa. Okay, so that points away from standard respiration.
Strongly. It indicated some kind of energy process that doesn't need oxygen, a non-oxidative pathway fueled by glutamine.
So where could that ATP possibly be coming from?
Their hypothesis focused on something called substrate-level phosphorylation.
Okay, what's that in simple terms?
Think of it like a quick, direct energy transfer step within a metabolic pathway, rather than the big, complex, oxygen-dependent machinery of respiration.
Like a shortcut for making a bit of ATP?
Sort of,
Yeah.
and they suspected it was happening within the TCA cycle, or Krebs cycle. Specifically, one step involving an enzyme called succinyl-CoA ligase.
So they thought glutamine was being processed partway through the Krebs cycle, and this one step was making a little ATP directly.
That was the idea, that glutamine could fuel this specific ATP-generating step, even without oxygen driving the whole cycle in the usual way.
Okay, clever hypothesis. How did they prove it? How did they confirm glutamine was going down that specific path?
They used a really neat trick, labeled glutamine. They used glutamine where the carbon atoms were a heavier type, C13.
Like putting a little tracker on it?
Exactly. So they could follow where those specific carbons ended up.
And where did they end up?
They found labeled C13 in succinic acid.
Succinic acid. Why is that significant?
Because succinic acid is basically a waste product or an end product of this specific way, cancer cells seem to process glutamine, the pathway called glutaminolysis.
Ah, so finding the label there means glutamine went through that pathway.
Precisely. And the fact that this succinic acid was being produced and sort of dumped out by the cells showed the glutamine wasn't being fully respired in the normal way.
It was being processed part way and then released, like fermentation.
Exactly. It fits the pattern of fermentation, taking a fuel, processing it partly for energy, and releasing an end product. In this case, succinic acid instead of lactic acid from glucose.
That seems pretty solid. Did they do anything else to confirm?
Yes, they wanted to be really sure. They did another experiment using cyanide.
Cyanide, isn't that like poison?
It is, because it specifically blocks the main oxygen-using part of respiration, oxidative phosphorylation.
Okay, so adding cyanide is like chemically removing the effect of oxygen on ATP production.
Right, so they added cyanide, still no glucose, but plenty of glutamine. And even with cyanide blocking normal respiration, they still saw ATP being produced, and they still saw succinic acid building up.
Wow, so that really nails it down. It's an oxygen-independent pathway using glutamine, making ATP directly and producing succinic acid.
It provides very strong evidence for that conclusion, yes. A second fermentation pathway.
So how does this change our view of, say, Otto Warburg's big theory about cancer metabolism?
It's a really interesting refinement, actually. Warburg was absolutely right that cancer involves a metabolic shift towards fermentation. He saw that clearly.
But he mostly focused on glucose fermentation, right, into lactic acid.
Exactly. That's what he observed and emphasized. What this new research shows is that he was right about the fermentation, but he didn't know about the second major fuel being fermented.
Glutamine.
Glutamine. So it's not just glucose fermentation, it's glucose and glutamine fermentation. Cancer seems to have this dual fuel fermentation strategy.
So Warburg had part of the picture, but not the whole fuel story.
You could say that, yeah. His core idea about metabolic dysfunction holds, but it's broader than he realized. These cells seem even more committed to fermentation than we thought.
And what about oxygen? Tumors do consume oxygen. If they're fermenting so much, what are they using the oxygen for?
That's a fantastic question this research raises. The common assumption is, oh, they're using oxygen for making ATP via respiration, even if they also ferment.
But maybe not as much as we thought.
Possibly.
Hmm.
Given how much energy they seem to get from fermenting both glucose and glutamine, the oxygen they do consume might be going towards other things.
Like what?
Well, maybe building biomass, making lipids, nucleotides, other essential components for growth. It opens up new questions about what that oxygen consumption actually signifies in a tumor.
Okay, so this is all compelling work in cell cultures, in the lab. Did they check if this holds up in, an actual living system?
They did, and that's crucial. They moved to in vivo experiments using mice with glioblastoma.
And what did they do in the mice?
They tried to target both fuels simultaneously. They used drugs to inhibit glutamine use, and they put the mice on a ketogenic diet.
Ah, the keto diet to lower the glucose availability.
Exactly. Induce nutritional ketosis.
Yeah.
So you're hitting both glucose supply and glutamine metabolism.
And the result?
A really powerful anti-tumor effect. The tumors slowed down significantly, and importantly, it seemed to have very little toxicity for the mice themselves.
Wow, so attacking both fuels worked well in a living model and was well tolerated.
Yeah, it strongly backs up the idea that these two pathways are critical in a real biological context, not just in a petri dish. The in vivo results really support the in vitro findings.
If this dual fermentation is so important in glioblastoma, what about other cancers? Could this be a general thing?
That's the big hope, and there's reason to think it might be.
Why?
Because we already know many different types of cancer show high levels of both lactic acid and succinic acid, those fermentation products.
So the metabolic signature is already there in lots of cancers.
It seems to be. It strongly suggests that this reliance on fermenting both glucose and glutamine could be a really common, maybe even universal feature across different cancer types.
Are they looking into that?
Yes. The researchers explicitly mentioned plans to investigate this in lung, colon, bladder, breast cancer, some of the most common ones.
That could be huge if it pans out. A unifying metabolic vulnerability?
Potentially, yes. It
Yeah.
could really change the game.
Now, you mentioned Professor Seyfried. He'd apparently suspected glutamine might be fermentable even before this study.
Yeah, he talked about that. His initial idea was actually sparked by some older research on diving animals.
Diving animals. How does that connect?
Well, a researcher named Hoka had found that when these animals dive and are deprived of oxygen, their tissues build up not just lactic acid, but also succinic acid.
Ah, the same byproduct.
Exactly. Hoka guessed it might be from amino acid fermentation, but didn't nail down which one. Seyfried saw that and thought, maybe something similar happens in cancer mitochondria.
That's a cool connection.
Yeah, and then he teamed up with Dr. Christos Chinopoulos, who's an expert on that specific energy step, the substrate-level phosphorylation in mitochondria. And apparently, this paper was the result of like 7 years of work.
Wow. Long haul.
Real dedication to nailing down the mechanism.
So if we step back, it's like the cancer cells themselves, through the waste products they pump out,
Yeah,
are basically telling us what they need. They're signaling their dependence on glucose and glutamine.
that's a great way to put it. The lactic acid and the succinic acid are like signals, metabolic signatures, saying we need these two fuels and we're fermenting them. We just had to learn how to read the signals properly.
Right. Now, what about this idea you hear sometimes that cancer cells are super flexible metabolically? That if you take away glucose and glutamine, they'll just switch to burning fats or ketones.
Ah, the metabolic flexibility myth, as Seyfried calls it.
So it's a myth.
He and his colleagues argue strongly that it is. Their work and other evidence they point to suggests cancer cells are actually quite restricted metabolically. They're heavily dependent on glucose and glutamine fermentation.
They can't just easily switch fuels.
They argue no. They've published other work showing cancer cells don't really thrive on ketones or fatty acids when glucose and glutamine are limited.
Yeah.
Their metabolism seems quite hardwired towards this dual fermentation.
That's a really important point because it challenges a common assumption.
It really does. It suggests they have an Achilles heel, metabolically speaking.
So this metabolic view, does it suggest maybe the metabolism changing is the first step in cancer, not the genetic mutations?
That's the paradigm shift this line of research points towards, yes.
Explain that a bit more.
The argument is that the switch to fermentation, driven by dysfunctional mitochondria, perhaps is the primary driver. It creates instability, inflammation, signals for growth.
And the mutations come later.
Yeah, the genetic mutations we see are more like downstream effects or consequences of this underlying metabolic chaos, rather than the root cause that kicks everything off.
That flips the conventional view on its head.
It absolutely does. It puts metabolism front and center as the origin point.
Okay, so if this metabolic picture is right dual fermentation of glucose and glutamine being key, limited flexibility, what's the therapeutic path forward?
Well, it points directly to a strategy of targeting both of those fuel sources at the same time.
Like the mouse experiment.
Exactly. Combining something like a ketogenic diet to really lower glucose availability with drugs that specifically block the cancer cells from using glutamine effectively.
Starve them on two fronts.
Precisely. Deprive them of their primary fermentable fuels. The idea is this could dramatically slow tumor growth, reduce the acidity and inflammation they create, and potentially kill the cancer cells selectively.
While healthy cells, which are more flexible, can adapt.
Right. Healthy cells can typically use ketones or fats much more readily, especially when the body is in nutritional ketosis. So you're hitting the cancer's vulnerability while potentially supporting normal tissue. Minimal toxicity is the goal.
So wrapping this up, the huge insight here is
It's really the confirmation that cancer isn't just fermenting glucose. It was relying heavily on fermenting glutamine too, through this distinct pathway involving substrate-level phosphorylation and succinic acid production.
this dual fermentation seems fundamental to their energy and growth.
Yes, a central metabolic pillar for them.
And for you, the listener, the takeaway is that this deeper understanding could genuinely pave the way for new cancer treatments.
Right, therapies that might be much more targeted at the cancer's core metabolic needs,
Wow.
potentially more effective and hopefully much less toxic than many current approaches.
It really offers a different kind of hope based on understanding the fundamentals.
It does, and it leaves us with a final thought, maybe something for you to mull over. If cancer cells have kind of been broadcasting this metabolic secret, their reliance on both glucose and glutamine through their waste products for decades.
What else might we be missing?
Exactly. What other fundamental truths about diseases might be hiding in plain sight, obscured by our current assumptions? Maybe looking at the metabolic roots of other conditions could unlock similar breakthroughs.
Food of thought, definitely. Makes you wonder what else is waiting to be understood on that fundamental metabolic level.